Hello, my name is Sam Nemiroff, and today you'll be learning about the risk of microbial keratitis from contaminated eyewash stations. One term you'll need to be acquainted with is free-living amoeba. So, what are they? Free-living amoeba are ubiquitous opportunistic pathogens found throughout nature, from lakes and pools to air conditioning fluid and even eyewash stations. And this is definitely a problem for potential users because if this organism comes in contact with damaged eyes, it can cause a potentially blinding infection of the cornea, known as amoebic keratitis, and it's extremely difficult to treat. What's worse, eyewash stations are prime breeding grounds for these organisms. When institutions fail to flush the warm standing waters from their emergency apparatuses, microbial buildup occurs, forming a bacterial biofilm which amoeba love to thrive in. In my study, I used culture-dependent and independent methods to analyze and confirm the presence of free-living amoeba in academic and commercial eyewash stations. For my culture-dependent methods, I identified amoebic presence using non-nutrient agar with E. coli. After plating and incubation, I checked the plate under a microscope, looking for tracking of isolates along the lawn of bacteria, indicating positive presence. For my culture-independent methods, I extracted DNA using a Helix extraction protocol. I then used this DNA in a multiplex PCR assay seeking to amplify a range of free-living amoeba. Gel electrophoresis allowed me to differentiate between different species of amoeba through comparison to included positive controls. The results of my study showed that 2/3 of the institutions I visited contained amoebic presence in at least one of their eyewash stations, and this was not a random occurrence. Furthermore, including molecular techniques increased my yield 1.4-fold, displaying that culture-independent techniques are indeed more sensitive than traditional culture methods. If institutions started to follow the 10-minute per week flushing guideline, amoebic levels in these stations would dramatically decrease, thereby dramatically decreasing the risk of microbial keratitis. In the meanwhile, potential users are at risk of a public health threat imposed on them by schools and other scientific institutions that rarely, if ever, maintain their eyewash stations.